Now let us take up the next favorite topic of examiner, apoptosis. You should apologize me that apoptosis is another dragging, creeping subject, all molecular level imagination. But maybe after apoptosis the movie will run a little more faster. Until then you should bear with me that we are a little slower. So apoptosis is a planned, programmed cell kill, as all of you know very well, and apoptotic cell will become round masses with intense eosinophilic cytoplasm, nuclear chromatin fragments. There is no necrosis in apoptosis, or inflammation of cells is not involved in apoptosis. It is a drop of the cell. And apoptosis is a mechanism by which programmed cell death will cause embryogenesis, etc., etc. And it does not elicit inflammation. Necrosis elicits inflammation, apoptosis does not. This is one point on which multiple times examiner will ask the same question. Examiner also, when he picks up apoptosis topic, he will try to prefer chalo, no inflammation, do the students know or not? Let us check. Standard questions first of all. Very rarely they make a special effort to create a question on topics like apoptosis. So you should know all standard questions about apoptosis first of all. And only when apoptosis become considerable in certain tissues, considerably it happens, then only it become apparent, because it occurs very rapidly, unlike a histopathological change of necrosis where slowly you can see neutrophils coming, lymphocytes going, macrophages coming. All these things you can say the gradation, not in apoptosis. So what are the important morphological hallmarks of apoptosis? There is a cell shrinkage, chromatin condensation, there will be cytoplasmic bleps and apoptotic bodies which will be classically be forming, and there is a phagocytosis of the apoptotic cells by the adjacent healthy macrophages, and the apoptotic cell will appear finally as a round oval mass with the intensely eosinophilic cytoplasm with a dense nucleus chromatin fragments, and that is how the apoptosis basically progresses. CD95 is a very important cluster differentiation protein which has a major role in apoptosis. Now we are going into one of the imaginative plane of apoptosis. Be very sure to imagine. Fundamentally, whenever there is a damage to DNA in a cell, it is destined to die. It is like in war, somebody had a very bad carotid rupture, multiple trauma, then what you will do? You will shoot him, because you can't anyway, you are sure that multi-organ failure and multiple trauma is there, there is no point in making him to further survive, so you will kill him. That is what we see in all the war movies, right? So apoptosis also, once there is a DNA damage, that cell is destined for apoptosis, and that is decided that it is going to be dropped. There are two ways by which the apoptosis is initiated on such a destined cell. One is called extrinsic pathway, other is called intrinsic pathway. Okay? So this FAS receptor is also called as a death receptor, and it is also called as CD95, APO1, TNF receptor superfamily member called TNFRS16. The various names are given to the same thing called CD95, which is called the death receptor. That is, a cell destined to die with apoptosis will typically express this CD95. And it is being found that it is a chromosome 10. The gene is located which will be responsible for the expression of the CD95 in those cells which are destined for apoptosis is what need to be remembered. And this forms what is called as a death inducing signaling complex called DISC, and such a cell which forms this complex then will undergo the apoptosis. So we will take up in the next question how this death receptor expression of it will initiate apoptosis. That is the next question we will take up. Now in apoptosis, APOF1 is activated by the release of which substances from the mitochondria? Basically, some of the genes will say carry on apoptosis. Some of the genes will decide don't do apoptosis. Accordingly, there are anti-apoptotic and pro-apoptotic genes fundamentally. And when the death receptor is expressed, then what will happen? FAS, death receptor, CD95 is then, once it got expressed, then what will happen? The anti-apoptotic genes are all silenced and pro-apoptotic genes are made to get active. So this fine tuning basically happens inside the cell. So we need to know that there are intrinsic and extrinsic pathways which lead to apoptosis. Expression of this death receptor, that is the CD95, is one method by which apoptosis is initiated. The second method is whenever a cell is destined to undergo apoptosis, the mitochondria of the cell become very leaky, and that will cause a release of what are called as SMACs, and release of this SMACs initiate apoptosis. That is another path, extrinsic and intrinsic path. There are the two paths. The mitochondrial leakage leading to release of the SMACs and that initiating apoptosis is called intrinsic, and the expression of the death receptor pathway is basically called extrinsic in apoptosis. Now let us talk about this intrinsic path where mitochondria are involved. Now once a cell is destined to undergo apoptosis, what will happen? The mitochondria releases a mitochondrial protein called SMAC. What does SMAC acronym stand for? It's called second mitochondria derived activator of caspases is the name given for it, SMAC. Then this SMAC is released into the cytosol with increased permeability of the mitochondria. Then SMAC will then go and bind with a inhibitor of apoptosis protein called IAPs, and it will deactivate them. And normally IAPs, what is the role of inhibitor of apoptotic process? The name itself says it prevents apoptosis. But the moment SMAC is released, it goes and binds with IAP. IAPs are inactivated and no more they can stop apoptosis from not happening. So apoptosis become initiated. So that is what basically happens. The inhibitor of apoptotic process IAPs, they normally suppress the activity of certain important cysteine proteases called as caspases. How does inhibitors of apoptotic process prevent apoptosis from happening? Caspases are the final proteases which cause the cell drop, and they are basically prevented from working by suppression caused on them by the IAPs. If IAP itself is suppressed, there is nobody to stop caspases, and caspases will go ahead with the apoptosis. That is what we need to fundamentally remember. Now there is one more thing. The mitochondria, the moment it become leaky, it will typically release cytochrome C, and the cytochrome C is another important mediator. Once the cytochrome C got released, then it will bind with what is called as APOF1 and ATP. APAF1 is a protein to which it will basically bind, and it will typically form a complex with the APOF1, that is cytochrome C, and the complex of APAF1 and the cytochrome C is basically called as apoptosome, and apoptosome in turn will cleave the pro-caspase to the active form called caspase 9, and in turn caspase 9 will carry on the apoptosis. It's all granny's story. The king went, prince went across 7 oceans, got the mermaid, and went into that small tunnel and finally got out the princess. So that kind of a story it is. You should imagine. And note some of the keywords. Caspase are the final mediators, and APOF1 promotes caspase activation after binding with the cytochrome C. And one more thing we need to remember is SMACs, which are released from the mitochondria, and they basically inactivate, not activate, the IAPs. Examiner, if at all frames a question, will only twist on these areas. He can ask SMAC will activate or inactivate IAPs. will IAPs inactivate or activate the caspase? So examiner knows English a little better than us, and we must be sure what is not and what is yes about inactivation and activation. So this is how the mitochondria releases cytochrome C, apoptosome formation, caspase 9 release. This should basically fit into your mind. Now, if you look at this SMACs, which are released by the mitochondria, there are some other things which also regulate the SMACs, because simply if mitochondria become leaky, it releases SMAC will come inactivate the IAP, and IAP inactivation causes caspases that lead to apoptosis. Means only one mechanism, if it is there, it is very leaky. There must be some supra commander which will say that, "Array, I wrongly ordered for apoptosis, let me stop that also." So there must be multiple regulation on this SMACs which should happen. So who will regulate it? There is something called BCL2 proteins, which is a family of anti-apoptotic genes. They will be able to promote or inhibit the apoptosis by regulating the SMACs is what need to be basically remembered. And the beautiness of BCL2 proteins is, even if cytochrome C got released by the mitochondria and it is very eager to bind with APOF1 and form apoptosome and then release caspases, the BCL2 command is more stronger. The BCL2 can stop apoptosis from happening even if cytochrome C is eager to initiate apoptosis. So mitochondria releases one cytochrome C, one SMAC. The SMAC regulation mechanism is more stronger to promote and stop the apoptosis more than the cytochrome C APOF1 associated mechanism on caspases is what you have to basically remember. So that is all the story of intrinsic pathway of apoptosis you should be very sure about. Now in situ DNA nick end labeling will quantitate what? All in year 2005 questions. Probably if you ask somebody who did 15 or 20 years of service in PHC, first of all to know what is apoptosis, that was not there when 15-20 years back Robbins pathology was there. But on the top of it, if you ask what is intrinsic extrinsic pathway, what is BCL2, activate or inactivate caspases, oh, that is all. Then they will say paper is very, I don't know whether tough or easy also, because I did not know whether I have written a veterinary science paper or MD paper. If 5 questions on apoptosis come, let us say, in a very bad dream, then your day is gone, I mean your year is gone, right? Because if you know, you will answer, no guesswork in the actual exam hall. Now, what is this in situ DNA end labeling? Basically, doctor, a cell which is destined for the apoptosis will have some damage in the DNA, and there are some nicks in the DNA. So, to quantitate the amount of apoptosis, how you can do that? All the cells destined for apoptosis, you discover them. How? Their DNA will have a intranucleosomal cleavage and breakage points. You discover that, oh, the DNA of this cell has very less number of breakages, the DNA has more breakages. Based on that labeling, you can know which cell is going to become apoptotic and the other which is not. How can you do that? We do and detect the DNA breaks by terminal deoxynucleotide transferase mediated deoxyuridine triphosphate in situ nick end labeling called tunnel is a technique by which you can be able to assay the degree of DNA fragmentation and can identify the cells which are bound to undergo apoptosis is what need to be remembered. Now we come to a very important question, which are anti and pro apoptotic genes. The BCL2 is a important gene whose protein is very important to regulate the SMACs. So the BAX gene, BAG gene, BAG gene, BOG gene, they are all pro-apoptotic, whereas BCL2, BCLX, BCLW are anti-apoptotic in nature. So you must know which genes are pro and anti. Favorite question, multiple times the same question will come. Then BCL2 is anti-apoptotic, it inhibits the apoptosis, and overexpression of this BCL2 is also bad. Why? If BCL2 prevents apoptosis means normally apoptosis is important for the tumor from not growing. Anything which shows tumorous tendency will be dropped down. Any mutation BCL2 will cause anti-apoptotic activity become excessive, and that will prevent apoptosis from happening, and those cells become cancerous. Excessive anti-apoptosis is also cancerous in nature. So that is how excessive BCL2 expression also leads to anti-apoptosis excessively, leading to malignant tendency is what need to be remembered. Programmed cell death is basically called as apoptosis, as all of you know very well. Now what genes basically inhibit the apoptosis? Once more a favorite question. BCL2 is anti-apoptotic in nature, and BCL2 family include BCL2, BCLX, whereas the BAC, all these genes, they promote the cell death is what need to be remembered. Now what are apoptotic bodies, doctor? Apoptotic bodies are formed by the fragmentation of the surface bleps which are on the apoptotic cell, and these membrane bound bodies composed of cytoplasm and tightly packed organelles without or with nucleus are basically called apoptotic bodies. Now you can see that once the cell got infected and it is bound to undergo apoptosis, what will happen? There will be a lot of membrane bleps, cell membrane bleps form, and these bleps will basically slowly condense and detach from the cell, and they form what are called as the apoptotic bodies. So what is the starting point for the apoptosis and programmed cell death to begin? It is the activation of the endonuclease which will create the DNA intranucleosomal breakage points, and the moment the intranucleosomal breakage points are created in the DNA, that initiates the process of apoptosis. So endonuclease is the first initiator by creating DNA breakage points for the apoptosis is what need to be remembered. So there is a cell shrinkage, rounding, chromatin condensation, etc., finally leading to the development of apoptosis. Now one of the steps at the time of apoptosis is, in the process of apoptosis, the nuclear envelope become discontinuous, the DNA inside it become fragmented, and this process is called karyorrhexis. And karyorrhexis is basically because of the endonuclease activity, which will cleave the DNA into short fragments which are regularly spaced in size. And this will create, if you do gel electrophoresis, this kind of a laddered appearance of DNA. Endonuclease is breaking it into multiple small pieces, and this intranucleosomal cleavage will lead to that laddered pattern. And DNA laddered pattern is one of the hallmarks of recognizing the presence of apoptosis, is what you need to basically remember. This is another favorite question of examiner. Where do you see DNA laddered pattern? Okay, are you able to see the ladder here? I hope you are able to appreciate the ladder here due to intranucleosomal cleavage. Now with regard to the apoptosis, there is an intranuclear fragmentation of DNA, caspase activation, and annexin 5 is an important marker of apoptosis. Now let us comment on this statement, which is a favorite question of PGI June. What is this annexin 5? Annexin 5 is a cellular protein, and it is very important generally for inhibition of blood coagulation because it will compete with the phosphatidyl serine binding sites of the prothrombin. Now when it is a competitor for the phosphatidyl serine sites, annexin, why do we use it in apoptosis? The reason is we use annexin as a probe. The annexin A5 affinity assay it is called as. It will detect the cells that have expressed the phosphatidyl serine on their cell surface, and only those cells which are undergoing apoptosis will express phosphatidyl serine. Whether they have expressed or not is identified by this annexin 5. So that is the reason annexin 5 assay is used to identify the phosphatidyl serine expressing cells, which are the apoptotic cells, is what you need to basically understand. The laboratory guys are very smart. They will recognize one property somewhere and use it as a label to recognize something else. That is a very good way of doing it, right? Now, what are the morphological changes in apoptosis? As you know, the membrane bleb formation, nuclear fragmentation, cell swelling, but there is no, membrane bleb formation and karyopyresis, they are the two things. No inflammation will be found is what need to be remembered. How does chemotherapeutic agents show their effect and kill the cancer cells? Two ways. Directly by initiating necrosis and apoptosis is one method, is done basically by the chemotherapeutic agents. And what do you mean by any? One of the terms given was anycosis. Anycosis is basically if you detach a epithelial cell from its cell adhesion, then that leads to death of it. If there is no cell adhesion, without company, cell will die, and that is basically called anycosis, not a part of apoptosis. Then next question of May 2007 was, everything is a feature of apoptosis, chromosomal breakage, clumping of chromatin, cell shrinkage, but not inflammation is not a feature, is what you should be very sure. Then once more, November 2004 AIMS question, which can be a potential question, the next state entrance is, ladder pattern of DNA on electrophoresis is basically because of endonuclease activity, which is responsible. First step in the apoptosis is what you have to basically remember. And apoptosis is classical hallmark is this internucleosomal cleavage of the DNA is very important. You can see in a real-time gel electrophoresis, the typical step ladder pattern created by this internucleosomal cleavage is what need to be remembered. Programmed cell death is what we basically call as apoptosis. Now once more question on anti-apoptotic and pro-apoptotic genes. The two main anti-apoptotic genes would be BCL2, BCLX, whereas the BAX gene, they are all pro-apoptotic in nature, as all of you know very well. Now the cytochrome C is very important for what? We discussed na? Intrinsic pathway, mitochondrial leakage. In the apoptosis, we require cytochrome C release is what need to be fundamentally remembered. Now we come to a very important question. We already discussed intrinsic pathway. Then what is left over for us? We have to still discuss CD95 associated extrinsic pathway. Now we are going to have a little once more a fairy tale about what happens in extrinsic pathway. Now fundamentally in extrinsic pathway of apoptosis, there is an expression of a cell surface death receptor in a cell destined for apoptosis. The death receptors are basically the members of the tumor necrosis factor receptor family, it is called as TNFR1 is what need to be remembered. And very well known death receptors are TNFR1 and FAS, which is also called CD95, TNFR1 and FAS. FAS is other name given for CD95. These two are the death receptors that we know very well. Now when the FAS receptors are cross-linked by its ligand, which is triggering the apoptotic event, the sequence become initiated. They basically will cause then the release of the active caspases. And how does that happen? Fundamentally, once this FAS got stimulated, it will typically will release what is called as a flip protein, and in turn will lead to activation of the pro-caspases into active caspases, and that lead to the development of apoptosis is what you need to basically remember. Only two buzzwords, FAS, CD95 and TNFR1 are the two things, and flip is another one involved in the extrinsic pathway of apoptosis. And what are involved in intrinsic pathway? Cytochrome C and EPOF1 and BCL2. That's all. Now, toll-like receptors, where do we find them and what is their importance in our regular antimicrobial activity? The toll-like receptors are the membrane proteins. They recognize the microbe-derived molecules. Once microbe is walking, there is a perfume which comes out of it. That scent is recognized by the toll-like receptor, and then it will stimulate the innate immune system against the microbe. So it is like a messenger. Oh, terrorist is coming, kill him. is what it will tell to the immune system, toll-like receptors. And where do we see toll-like receptors expressed? In different cell types that participate in innate immune system, that include macrophages, dendritic cells, NK cells, neutrophils, etc., etc. And how will toll-like receptors, fine, they will recognize that there is a microbe which has come. How will they go and tell innate immune system? There must be some mediation in between, right? So they cause the transcription factors called NFKB and AP1, they become stimulated, and in turn they will go and they will basically cause the innate immune system active, and then that will cause the microbial killing is what need to be basically remembered. And the toll-like receptors are also important for initiating inflammation and response of it by causing cytokines, E-selectins, etc., to become expressed excessively at the onset of inflammation. Now the simple question of November 2007 is, caspases are important for apoptosis. Now you are very, very sure about. That is piece with the topic of apoptosis.